Remember the first function was GFR, to filter. At the corpuscle, filtrate is produced. Things are excreted from the body, and then we reabsorb and secrete in the tubules as we go through. So GFR is fairly high, and it's fairly stable. We discussed the mechanisms that regulate GFR and the factors that result in GFR. So there's a pressure inside the glomerulus that pushes fluid and solutes out. There's a pressure in the capsule already that opposes that flow. And there's colloid pressure from proteins that try to attract those solutes and fluid as they leave. So the net result of these three factors dictates if and how much filtration occurs. And we want to keep this constant within tight ranges. We achieve that mainly through autoregulation. So two main mechanisms of autoregulation: myogenic mechanism, where stretch in the afferent arterioles causes a reflex to oppose that stretch. So if they're stretched, the afferent arterioles constrict, and that limits flow into the glomerulus. If they're not stretched, then they open up to encourage flow into the glomerulus. Both of those cases ensure relatively stable GFR. And then we have the ability through the juxtaglomerular apparatus to sense how efficient our efforts have been. What are the concentration of solutes leaving the distal tubule? And we communicate how efficient we've been back to the afferent arteriole to adjust accordingly, affect filtration. So this is what we covered last class. Between GFR in the glomerulus and the apparatus in the distal tubule, we've got a whole series of events that describe reabsorption and secretion. It's harder to separate these two into distinct items, so we're talking about them together. You already know that a lot of the filtrate is reabsorbed. The large majority is reabsorbed. And good thing, too, because here are some examples of amounts of solute that pass out of the glomerulus into the tubule. Water is reabsorbed, most of the glucose, almost all of the glucose is reabsorbed. Sodium, potassium, calcium, a whole host of other things too. And these numbers, sodium filtered out, 579,000 milligrams filtered out of the blood at the glomerulus. Most of it's reabsorbed, some is excreted in the urine. Your RDA for sodium is in the 2,000 milligram range. If you have 3,000 milligrams in a day, that's a red flag. If you do that a lot, people think that leads to high blood pressure. This is 100 times that amount or more, 200 times that amount. Large amounts of sodium filtered out. Same with potassium. Glucose completely reabsorbed. And the proximal convoluted tubule is the major site of this reabsorption. Not the only site, but the major site, the majority. Over 50% of these elements reabsorbed in the proximal convoluted tubule. And so this absorption, this return back to the blood, is relatively automatic. We can change it in the later portions of the tubule. We fine-tune or titrate the amounts that are being reabsorbed because we have control over some of the mechanisms later on in the tubule. A little bit at the nephron loop, and we'll go through a detailed example of the nephron loop and the countercurrent mechanism tomorrow. And then in the distal tubule, we have control over the re-entry of some things back into the blood. We can either encourage them or not. And we'll cover that at the end of the lecture today. We saw that in lab 8, the urine lab. So reabsorption, a major focus. Secretion is less of a focus, but equally important. Small amounts of things are secreted, but usually they are either toxic or severely detrimental to the body and need to be excreted. Breakdown products of heme, for instance, breakdown products of amino acids, creatinine, which is a breakdown product of creatine in the muscle, excreted through the urine. Some different hormones, drugs, cocaine, alcohol, other metabolites, excreted in the urine. So you can learn a lot about a person's physiology from determining what substances and in what amounts are being excreted from the body. And it's not a quick process. A lot of these things will hang around for weeks afterwards. So often toxic substances in smaller amounts that get secreted. The large focus of this section is on reabsorption, but note some things are secreted. How do these move between the tubules, back through the interstitial fluid and into the blood? How do they move? Either between cells or through cells. And movement between cells is shown here, described as paracellular reabsorption. This could be water, this could be ions. It depends on how tight the junction is between cells. If there are leaky junctions or if there are tight junctions. Remember that from week 1 or 2 in the first semester, the different kinds of junctions between cells. This is typically passive. If there is a space, if there's an opening, things can move. You don't often have pumps or channels between cells. The active forms of transport here don't apply. Those do apply when you move something through a cell. The tubular cells are those that line the tubules. We'll see different kinds when we get to the distal convoluted tubule, but we'll think about them just as cells for now. And like some cells in the small intestine, they have villi to increase the surface area, they have pores, they have channels, they have pumps. And when we move things through the cells, it's called transcellular reabsorption. Here, antiport, symport, active transport all apply. These are the main routes of reabsorption. You can see it here. Pretty straightforward. In both cases, sodium is moving, water can move, potassium can move, although it's not as linear. Chloride can move in these, or through these mechanisms in this fashion. I don't need to spend a lot of time on this. You already know the different kinds of transport. We spent time last week introducing it. We covered it again on Tuesday. You know simple diffusion is a process that involves the movement of solutes and it's unaided by proteins or pumps. The primary active transport requires a pump and energy. Sodium potassium ATPase is the golden example, the gold standard example that we always refer to. ATP is broken down, energy is used to achieve this transfer of sodium and potassium. We'll talk about that today because that makes the sodium gradient, or it accentuates the sodium gradient, and then leveraging that sodium gradient allows us to move other things as well. And we move those other things through secondary transport. When we have a large gradient for one thing that moves downhill, we can leverage that large gradient to move another substance uphill against its concentration gradient. And you know symport are those things moving in the same direction. Antiport in opposite directions. So we'll come back to these, just a brief reminder of what we'll see today. Now these describe movement of solutes, typically ions. They don't describe movement of water. Water movement is generally passive, and there are some structures that will permit water to move more easily, but water movement is generally passive. Water moves because we will actively transport ions, and we want the total concentration of fluids in the body to be stable or unchanged, quote unquote normal. And by moving those ions, we're disrupting that balance. When we actively move ions from one compartment to another, the destination becomes more concentrated, and we don't want that. So water moves in order to maintain a normal concentration. In the kidneys, here's my rudimentary nephron. As fluid comes in, it's called tubular fluid at this point. The obligatory water reabsorption that just happens automatically due to normal movement and pumping of ions largely occurs in the proximal tubule and accounts for 90% of water movement. Water is drawn by osmosis, it moves passively, doesn't need permission, but we can change how easily water moves later. We have control over whether we reabsorb a lot of water or slightly less water. And that describes facultative water reabsorption, which occurs mostly in the distal convoluted tubule and in the collecting duct, where after all the processing has taken place, tubular fluid becomes urine, and we can modify the size of this reabsorption if we have signals to do so. One of them that you've seen before mentioned in lab is ADH, antidiuretic hormone. We can modify how much fluid is reabsorbed by giving it permission to be reabsorbed. So let's talk about the mechanisms in each of these areas. This is a broad overview to start. What are the details of this movement? Proximal convoluted tubule, the first stop of tubular fluid after it's been filtered. The filtrate is new, nothing has been reabsorbed yet. It enters the proximal convoluted tubule where 2/3 of water, sodium, potassium, major solutes are reabsorbed. Large majority of the filtrate is reabsorbed in the proximal convoluted tubule. And because the filtrate coming in is fresh and there are high concentrations of things like sodium, 580,000 milligrams in the filtrate coming into the proximal convoluted tubule, we have a large sodium gradient that we can leverage to move both it and water and other substances. And so the mechanism that's on display here is of sodium-assisted glucose transport. But this approach is used for amino acids, it's used for other ions, and when we actively move sodium out, water will follow. So largely, reabsorption here is driven by sodium-driven symporters. So you're seeing glucose and sodium move together. Because there's this large gradient, so much sodium has been filtered out. There's little in cells. It's a normal cell in the body. You know, sodium concentration is precipitously low inside the cells, and we keep it that way. There's a large gradient across this membrane, and glucose hitches a ride through this symporter. It normally wouldn't, it's an energetic process, but it can hitch a ride because the gradient for sodium is so large. Sodium gets expelled through the normal sodium-potassium pump. Sodium-potassium ATPase. It's in all cells, always working. And then glucose it diffuses out, facilitated diffusion out into the bloodstream. And it's through this mechanism that all glucose is reabsorbed. So symport, leveraging the sodium gradient, is responsible for a lot of movement of these solutes. what else is there in the proximal convoluted tubule? Movement that doesn't follow that same blueprint. Slightly different movement. Here we see antiport of protons, hydrogen ions. Sodium can be reabsorbed again along its concentration gradient in exchange for hydrogen ions being expelled from the body. We have our first example of secretion in this case. So sodium will want to move into the body, again because of the large gradient. And using this antiporter, it can do so, but only if there is a source of protons present. Through this specific transporter, sodium can move in only if there are protons present to move in the opposite direction. If there aren't protons present, that doesn't mean sodium won't move. It just won't follow this avenue. Sodium will move through other means, like the glucose transporter that we saw on the last slide. But this specific mechanism to help control pH in the body will only allow sodium to move if there are protons present. So we need a source of protons inside the cells. Otherwise, this antiporter won't work. And we get protons from our old friend, carbonic anhydrase reaction. CO2 and water combine to make carbonic acid that dissociates in normal neutral pH to protons and bicarbonate, providing a continual source of protons for this antiport mechanism. And accordingly, we can help regulate pH. By doing this, we expel some protons, bring pH back up, and neutralize the potential acidic conditions in the body. So, how do we keep this going? Bicarbonate has to leave, so this reaction can keep moving forward. If not, it will stall. Sodium movement won't continue. Bicarbonate moves out, protons move out, and conveniently enough, a secondary effect of this mechanism, we have absorption of sodium, we have secretion of protons, and a secondary mechanism, a secondary effect, is that we reabsorb bicarbonate that had been filtered out into the tubular fluid. Protons leave, carbonic anhydrase in the border of the proximal convoluted cells again creates CO2 and water. CO2 can move back across, gases diffuse easily across the membrane, and the cycle continues. Now this looks circular at first. Why do all this work just to have everything return to normal? You're not getting anywhere. Let's follow the carbons. Bicarbonate, movement of bicarbonate you can really see when you follow the carbon. So bicarbonate moving in follows this general scheme where CO2 enters the cell, recombines with water, but diffuses out into the blood. So this is complete reabsorption of bicarbonate. And similarly, you can see the excretion of protons, which would be sourced from water initially. They're expelled through the antiporter and continue in the tubular fluid or excreted as urine. So it's not circular, even though the reaction happens in both compartments. There is net movement of bicarbonate in, net movement of protons out. The large movement with the sodium gradient. Sodium can help control acid-base balance in the body through that mechanism. What does the implication of this movement suggest? If we have this large gradient for sodium movement, what does that allow to have happen afterwards? If we start to pump sodium from the tubular fluid back into the body, we leverage that gradient to accomplish some work, we're altering the osmolarity on either side of the tubule membrane. Remember, we want to keep osmolarity rather consistent. And so if we continue to pump things back into the body and reabsorb, water will follow passively to equalize that concentration. So we have a movement of solutes that we just described in the last two slides. Osmolarity is increased on the right-hand side of this graph. That means water moves passively through both transcellular and paracellular routes to satisfy or neutralize, to bring back into balance the concentration of fluids within the body. We have our first inkling here as well, and I only bring this up to not shock you when we talk about it more in depth later, but the permissive ability, the ability to give water more of a chance to move is shown here through the actions of aquaporin 1, which is a channel specifically for water. Water will move through membranes passively, but it can do it more easily if there's an opening. And aquaporin, the aquaporin group of channels, is a specific water channel that allows water to move more easily between two areas. So we have aquaporin in this area that allows water to move, it facilitates water movement. And we can change the amount of aquaporin that is present to change how easily water moves. This is more obvious later as we get to the distal convoluted tubule. But just note, like with other ions, there are pores, there are channels for water. It doesn't need them necessarily, but it does help to facilitate that movement. So there are a number of mechanisms, a number of transporters, and we're only looking at a high level, but the high level involves sodium movement first and foremost. There's a large gradient. Things that move in conjunction with sodium, either because of primary active transport and sodium being pumped out, or by things hitching a ride against their concentration gradients, leveraging that large sodium gradient. Glucose moves, as well as amino acids, lactate, sulfate, phosphate. They move in unison with sodium. They move in the same direction with sodium. They are reabsorbed via symport. Bicarbonate moves via mainly facilitated diffusion and as part of the antiport system that we discussed in a little bit of depth earlier. These things being actively transported from the fluid back into the body create the environment for other elements to move. Specifically, water will move passively to follow the concentration gradient. And without going into too much detail, we create a concentration gradient for things like chloride, magnesium, and potassium to move as well. The detailed approach to ion movement in the kidneys, and the nephron specifically, is almost endlessly complicated. This seems complicated, but there's much more detail that we're not looking at. Largely sodium driven, we create the conditions for the movement of other things. This summarizes the role of the proximal convoluted tubule. And at that point, tubular fluid enters the loop of Henle. The loop of Henle has a large role to play in the countercurrent mechanism that we've heard mentioned of before, but we don't know details on yet. That's coming tomorrow. As the tubular fluid enters the loop of Henle, a lot of the heavy lifting's been done. 2/3 of the filtrate has been reabsorbed. There's no glucose anymore. There are few amino acids and proteins, if there were to begin with. 65% of the filtrate absorption has been completed already. Water's been reabsorbed as well to equalize concentrations, which is why it's still of a similar composition to blood. If you were to measure blood and look at the concentrations of various ions, it would be similar in the blood as in the fluid entering the loop of Henle. But this is a special compartment because it is impermeable, or partly impermeable, to water. So far, movement of ions will necessarily mean water has to move as well. In the loop of Henle, movement of ions can be divorced or uncoupled from the movement of water. Some water can be reabsorbed in the long descending loop of the loop of Henle, or the descending arm, but the ascending arm is essentially impermeable to water. It's not impermeable to things like sodium or chloride, where those ions still are moved out. We still have movement of some ions. We want to equalize concentrations. The body wants to maintain normal osmolarity in all compartments, but here it can't. Water can't move. And so the consequence is that while we continue to pump ions out, we get an imbalance in concentration between the tube and the cells or the body around the tube. And this is really important in understanding the production of urine, either concentrated or dilute urine, when we discuss that example in depth tomorrow. So water and ion movement are not coupled. They can be divorced here. So I want to show you the movement, the ion movement that I described, and this is independent of water movement. You guys all got this? Okay. I heard some dissatisfied groans. The mechanism by which we pump ions out is shown here. A lot of things happen in unison together. Sodium, potassium, chloride, all moved from the fluid, out of the fluid, into the cells via one symporter. A sodium, potassium, chloride symporter. Sodium and chloride happen to be reabsorbed. Potassium is not. Potassium it's weird, it's fickle. It leaks back into the fluid, and it sometimes leaks into the blood, and it's really hard to tell which way it's going to go, because there are openings, and it's not on this figure that you can see it, it's in one of the ones coming up. There are openings in both directions. And it really would just depend on the concentration gradient, but your guess is as good as mine as to which way it goes in any given situation. Sodium and chloride are reabsorbed. Potassium leaks back out. So we have this movement of ions that does not prompt the movement of water. I wanted some animations to come up before this last point. I'll come back to this point in a second. So we have all of these ions that still remain in the tubular fluid. Most of them have been reabsorbed. 2/3 have been reabsorbed. But there are sodium, potassium, chloride ions in the tubular fluid. And these are pumped into the cell via this symporter. I shouldn't say pumped. Pumped implies energy requiring process. They are moved, or they move into the cell via the symporter. The energy comes from, again, the sodium gradient. So these move into the cell. Sodium is pumped out of the cell, here using ATP, using an active transport. The ATPase pumps sodium out where it is absorbed into the blood, and then chloride follows along. Chloride follows along because its negative charge is drawn to the positive charge of sodium. Potassium doesn't play along, and potassium loops back out to be moved again, and this is a futile cycle. This is an example of circular movement that gets nowhere and accomplishes nothing. Potassium moves in, it leaks back out. It moves in, it leaks back out. Sodium and chloride are reabsorbed, potassium is not. But it doesn't mean it has no role to play. Always leaking potassium back out creates a situation where there's a higher positive charge inside the tubule than in the cells, than in the blood. And it's this new situation that's created by a movement of these ions that helps to push other solutes out. Calcium, magnesium, sodium all have positive charges, and they are pushed out, they are assisted in their reabsorption via the leakage of potassium. So all told, we have fairly convincing movement of ions here, but no water movement. High throughput of ions, but no water movement. We get sodium, we get calcium, we get magnesium movement. Chloride moves as well, but water does not follow suit. And so what happens, unbeknownst to us, it's outside of our control, concentration goes up. There are more ions in the same amount of space. And this is the basis for the countercurrent mechanism that we don't know anything about yet.
Osmosis passively draws water out of the proximal convoluted tubule and descending limb of the nephron into the peritubular capillaries. The ascending limb of the nephron loop is impermeable to water, but actively pumps sodium and chloride ions into the interstitial fluid. The resulting buildup of salt surrounding the nephron loop draws about 80% of the water from the descending limb into the interstitial fluid and into the capillaries, regulating fluid volume and osmolarity. This process of water following the solutes is known as obligatory water reabsorption. Some nephron loops extend deeply into the renal pyramids, allowing a process known as countercurrent multiplication to take place. Countercurrent multiplication forms and maintains a vertical osmotic gradient which allows additional water to be reabsorbed from the distal convoluted tubule and collecting duct. This controls plasma osmotic pressure and concentrates urine.
So you're not supposed to know what this is yet, but we're setting the foundation for exploring this idea further tomorrow. Vertical osmotic gradient will somehow prompt fluid to move. So this is nothing new. We saw this video the first day that we got into the section. This is all a summary of the information that we just presented. Ions move out, water doesn't, and we create this inequality between the loop and the cells surrounding it. So this inequality continues into the last part of the nephron, the distal convoluted tubule, where we finally have control over some of the fluid being reabsorbed. We can vary the amounts through hormonal signals. And at this point, 80% of everything that was filtered out has been reabsorbed. 80%. So we have 20% to play with in the distal convoluted tubule. We can either shut off reabsorption and 20% gets expelled as urine, or we can send the cavalry in, alarm bells are going, reabsorb everything, and maybe only excrete 1 to 2%. You'll never not excrete something. But 80% has been reabsorbed as we enter the distal convoluted tubule. Here, the first thing that happens, perhaps unsurprisingly, sodium and chloride are moved in, are reabsorbed, which will pull water with it. In this stage, we're outside of the loop, so there is water reabsorption. But we have control over the activity of these symporters through the actions of aldosterone. Aldosterone is a hormone that I'll refer to coming up when we talk about hormonal regulation again, that can accentuate this activity. Normally, sodium and chloride move in, and then 10% of what's left will be reabsorbed. Interestingly, this is one example that doesn't deal with sodium, so I thought it was worth including. Cells in the distal convoluted tubule are also sensitive to parathyroid hormone, which has an influence on how much calcium is reabsorbed. Depending on the body's needs, parathyroid hormone will be released from the thyroid gland and can exert influence here to reabsorb calcium if the body's stores are low, or to not reabsorb calcium if they're high. But it's an example of non-sodium and non-chloride related movement that I thought was interesting to put out. So we have the opportunity here to modify sodium reabsorption and thus fluid reabsorption through the actions of aldosterone. As we proceed through the distal convoluted tubule, we add to that control by modifying our ability to reabsorb water. Later on, after we've exerted that control with aldosterone, and perhaps the average 10% of filtrate has been reabsorbed, there's only 5 to 10% left. 90 to 95% of all of the filtrate has been returned to the blood. Glucose, amino acids, ions, fluid. The large majority has been returned to the blood. We can further titrate or modify the reabsorption in the distal convoluted tubule through actions of hormones on one of the two major types of cells in the distal convoluted tubule. Until now, we've just considered tubular cells as cells. There are two major types of cells in the distal convoluted tubule, one of them being principal cells that are outlined here and shown in the diagram on the right. And principal cells are, again, a site where we have control over sodium reabsorption and where we're introducing control over water reabsorption. These cells are responsive to hormone signals from aldosterone and antidiuretic hormone. So their normal actions are shown here. Like any cell in the body, the sodium, potassium, ATPase maintains our ideal conditions. Low sodium in, high sodium out. High potassium in, low potassium out. That's the job of this pump. And this pump is always working to keep the status quo, but we have potassium and sodium leaking through from the tubule. Sodium comes in, is expelled. Potassium returns to the fluid. So this transporter, this pump, can be activated by aldosterone. And the effect of that activation is reabsorb more sodium. If there's a situation where we need to reabsorb sodium, we would do that by aldosterone making this work faster, put it that way. What's not shown here, that I'm going to add in using my great PowerPoint skills, are aquaporins. the water channels, similar to the family of water channels that we looked at initially. Are they the same? I think it was aquaporin 1 the first time. Aquaporin are channels that can be inserted in the membrane that allow water to move through. And these things normally sit inside, disconnected from the outside of the cell, not doing anything. When we need to reabsorb more water, these move from inside of the cell to be inserted in the membrane, and when they're inserted in the membrane, they allow passage. They make a portal for water to go through. So we have control over how much water is reabsorbed when ADH induces aquaporin to be inserted in the membrane. Those signals vary depending on blood volume and blood pressure and blood osmolality, and it was one of the major objectives of Lab 8, the urinalysis lab, to try to get a sense of what signals will cause more or less fluid to be excreted, more or less sodium to be excreted. How might these hormones be affected to accomplish that goal? And I'll introduce, we should have time, I'll introduce what my take on that is coming up. The second type of cell isn't involved in sodium reabsorption, isn't involved in water reabsorption. It's instead involved in acid-base balance. And I'm purposefully limiting our discussion on acid-base balance here. It's not controversial, but I'm trying to decide whether this is an important enough concept to bring back up in class tomorrow or next week, given our limited amount of time left in the course. I love this topic, but we still have a lot of other subjects to get through. So the intercalated cells are involved in acid-base balance, where they can reabsorb bicarbonate and secrete protons, H+, to reduce acidity. And they're one of the only sites where we get a significant reabsorption of potassium. Until now, potassium's just been leaking out, not being reabsorbed. And if that were the case throughout the nephron, eventually run into a situation where you might have low potassium in the body. So the intercalated cells are important in reabsorbing potassium. It's one of those sites to make note of. So I'm not going to go into depth on this for now, but maybe later. We'll see. So regulation of sodium and water. You know this negative feedback loop well already. ADH, we talked about in, I forgot to look up the set of slides, but it was in the first semester. We're in the pituitary gland. We're in the hypothalamus looking at adrenocorticotropic hormone. I think it was in the endocrine section, right at the end of semester 1. And we noticed the hypothalamus was bathed in fluid that mimicked blood composition. And it sensed directly that fluid, and if osmolarity changed, the hypothalamus could release antidiuretic hormone directly from the secretory nerves in the hypothalamus to affect a change of the kidneys. So the hypothalamus senses blood osmolality. If it goes up, if it gets more concentrated, it's not a good thing. We want to maintain a stable concentration always. So the hypothalamus senses osmolarity directly. If it goes up, that's a signal to institute a change and correct that high osmolarity. So ADH is released, travels to the distal convoluted tubule, where it promotes the aquaporins to insert in the principal cells. Water can be reabsorbed. Consequence of that is water is reabsorbed, which is good. Reabsorbing more water will dilute the fluids of the body, so concentration goes down. We have higher volume, lower concentration. So we reinstitute the normal situation. Now I want to propose a scenario. So this outlines what happens if blood volume is too low. This is one of the elements that would happen if blood volume is too low or blood concentration is too low. We sense that we need to reabsorb water somehow. How do we get this response? How do we integrate this response from a larger whole body point of view? Let's imagine a situation, dehydration, severe blood loss, hemorrhage, where blood pressure is precipitously low and we are in danger of fainting, of passing out, of losing flow in the vasculature. What would be a typical response to that situation of low blood volume, reduced blood pressure?
Hormonal control of water retention.
That's not supposed to go on there.
Hormonal control of water retention also occurs in the nephron. Extracellular osmotic pressure.
All right, you know what? I'm going to fix these animations. We're close enough that I'm not going to get through it, so let's call it there for today. We'll pick this up as a fresh example tomorrow. Before we get into the countercurrent mechanism, have a wonderful Thursday.